in Europe, and good morning to all our colleagues in the US who are linking in. My name is Ilan Chaitovitz. I am an investor with a healthcare background, but more importantly, I'm also treasurer for CRDN, the fantastic charity that is hosting this wonderful event today. I'm really excited to be helping out with this afternoon slot. It's going to be looking at medicines discovery, and in particular, the potential for genomic medicines within the rare disease space. On that note, I'm very happy to introduce both Steve Rees, who is the VP of Discovery Biology at AstraZeneca, and also Sharon Barr, Head of Rare Disease Research and Development at Alexion. So wonderful to have you here with us. Thank you so much for your time, Rees, Steve, and Sharon. And I was just wondering if you could just tell us a little bit about how you do your day job, what you are involved with, and what that means in terms of bringing drugs from the clinic to the patient. Steve, do you want to go first?
You go first.
We're both being very polite. So thank you, Ilan, and huge thanks to CRDN for inviting us to talk this afternoon, and thanks to everybody for joining the session. Yes, so as I say, my name is Steve Rees. I lead a department called Discovery Biology at AstraZeneca. I've been working in the pharmaceutical industry for over 30 years now. Throughout that time, working in early phase drug discovery. So in my day job, what we do is we discover the targets, new targets. So we ask the question, which proteins, which receptors potentially should we target that would lead to the creation of medicines that would have efficacy in disease? And then we're involved in developing the reagents and the assays that allow us to find the first molecules that will ultimately become the medicines that are in the clinic. In recent years, we've become very interested in gene therapies, and that's really why I'm here. We've done a lot of work to create new methods inside AstraZeneca that allow us to create gene therapies that potentially allow us to cure disease rather than treat disease. And what I'd like to do today is talk a little bit about how we're using those approaches and now applying those approaches potentially to allow us to address rare diseases.
Wonderful, thank you so much. And Sharon?
Yeah, hard to follow up with Steve. Steve and I have very similar jobs, which is why we get along so famously. So I am the Senior Vice President, Head of Research and Product Development at Alexion, AstraZeneca. Until very recently, I worked for Alexion, which is a biotech company that focuses on creating therapies for patients with rare disease. And a little earlier this year, our company was acquired by AstraZeneca, which I think has created a new world of opportunities for creating therapies for those patients. So we're very excited about this acquisition. We think it's a very positive thing for our company, but more importantly for the patients that we hope to serve. Very much like Steve. Oh, sorry, go ahead.
Sorry, I didn't mean to interrupt you, but it was actually a really good segue. Firstly, into why we've got both you guys on the chat today, but also you mentioned that you're very excited about this deal. I'm very excited about it, but I'd love to hear from you, Sharon. You mentioned what gets you revved up about the combination of these two amazing companies.
Besides the chance to work with Steve. Working at Alexion has been a really fantastic experience because our whole endeavor has been so completely focused on delivering therapies for patients in need. But where there's a will, there isn't always a way. We were a smallish biotech company, and we are good at making large molecules. We're good at making antibodies and sometimes enzyme replacement therapies. And we have some ability to make small molecules, although it's very, very limited. And so at a certain point, we reach a barrier where we have a desire to create a therapy, but we don't have the technology or the platform to do it. And so that can be a little frustrating from a scientific perspective, to understand the science that you want to go after and the kind of therapy that you want to build for patients, but you don't have the capability of doing it. And so with this acquisition, we walk into a much larger company that has invested very heavily in building platforms and capabilities that are necessary for making these new therapies. And those are platforms and capabilities that for a long time, we at Alexion Research wished that we had, but we didn't. And so starting on July 21st, suddenly it blows the doors off of research for patients with rare disease, because we have the chance to work with experts like Steve and his colleagues throughout the organization to invent therapies that we weren't quite able to attack before. And I think that's really interesting for scientists, but more importantly, very valuable for patients with rare disease.
Sharon, thank you so much for a really erudite explanation of why you're so enthusiastic. That makes a lot of sense to me, but there's a lot of talk of genomic medicines and rare diseases, but could you just explain, or one of you please explain, what are genomic medicines and why in particular are these relevant to the rare disease space?
So I think maybe if I go first and then Sharon, please do add. And the first thing actually I'd just like to do is just back up a little and add to your comments. Alexion has been part of AstraZeneca for barely 2 months now, and the collaboration and partnership has been outstanding. And the opportunities in which we can bring AstraZeneca capabilities to bear to develop new treatments for rare diseases, I think are also immense. And much of our choices now are where do we choose to work? What are the diseases we choose to progress? So within AstraZeneca, as in any large pharma company, we've built a range of technology platforms that allow us to develop all different sorts of medicine types to treat disease. And historically, our focus has been it's cancer, it's heart disease, respiratory disease, and the like. We're now able to bring those technologies to bear to try and develop treatments and maybe cures for rare diseases. And when we're talking about genomic medicines, what we're talking about here are medicines that allow us to correct genetic defects that lead to rare disease. And as everybody around this table knows, many rare diseases, if not most rare diseases, have a known genetic defect. And that allows us to take technologies like gene therapy, CRISPR therapies, and oligonucleotide therapies to develop treatments that we believe give us have strong potential to treat these diseases, maybe even cure these diseases using these new medicine approaches. And one of the big advantages from a drug discovery perspective of working with a rare disease, as opposed to working with some of the more common diseases, is that at a genetic level, the cause of the rare disease is normally very, very well defined. So to a greater or lesser extent, we know what we need to do to be able to treat that disease. And the challenge then becomes creating a medicine that delivers that efficacy and also is safe, that allows us to take that to patients.
That is fantastic, Steve. Thank you so much for that addition. That explains it, very, very clearly to me. But then you're left with the real question of where do you go from there? Which diseases do you focus on? Sharon, would you take that one?
Sure. So, in the world of rare disease, we spend a lot of time thinking about this, which are the experiments that we really can do. And when we started collaborating with Steve and his colleagues, we started at the very beginning. How many rare diseases are there in the world? Well, there's at least 7,000. And so we call that the rare disease map. We started from there and we filtered down and we said, okay, so how many of those 7,000 diseases have a known causal mutation? Do we understand the underpinning of that disease? And then of those, how many of those are diseases where we think it's possible to create a therapy? So if the disease results in a severe congenital malformation, there's really nothing that we can do that's going to reverse that after birth. So there's some things that we won't be able to address with the therapy. So that takes a few things off the list. And then we move to things where we understand the cause, we understand the prevalence, how many people roughly are there in the world with this disease, and we understand the nature of mutations. They're somewhat characterized. We know what genes they happen in, we know roughly how many mutations there are. And now we know how to go about that. So we can understand how technically feasible it is. The next more complicated part of the question is, if we're going to deliver a genomic medicine, a CRISPR or a gene therapy, where are we going to deliver it to? If it's a disease in which we deliver the therapy to, for example, the liver, because the protein will be produced from the liver, as many proteins are, scientifically, we understand how to do that. We understand how to deliver a new therapeutic to the liver. But if we need to deliver that therapeutic to somewhere else, for example, the brain, that's much more difficult. So we focused on the things that we know how to do today. For example, delivering therapeutics to the liver where we can produce the corrected protein. And we agreed that we would start building the technology so that we could get to the more scientifically challenging diseases next. So we thought a lot about what we do now based on what's scientifically feasible today and what we want to do tomorrow and how we have to build our technology to make that possible. Does that answer your question?
That's great. Thank you, Sharon, for that. But you did bring into play a bit of jargon, which I want to get a bit of clarity on, which is CRISPR and CRISPR medicines. And there's been a lot of talk about over the years. If you read any scientific news article or rag, you're going to come up against that term. But it's not clear to me still exactly what it is. And as Steve, would you go into a bit of detail as to what CRISPR is and what it's enabling you guys to do?
No, absolutely. And from a scientific community perspective, CRISPR is potentially the most exciting development in the last 50 years. CRISPR was only discovered in 2012, so 9 years ago now, just 9 years ago. And what CRISPR allows you to do, theoretically, is to change the sequence of any piece of DNA in the genome. It allows you to delete a gene or it allows you to repair a gene. And that has massive potential across a whole variety of different areas, including medicines development. And these medicines are in the clinic today. There are phase 1 clinical studies underway today using CRISPR. And the examples are in the rare disease space. So CRISPR Therapeutics is working with a company called Vertex to develop treatments for sickle cell disease and beta thalassemia. And this involves taking blood cells from these patients. It involves correcting the gene in those blood cells and reinfusing them back into patients. And the first patients on those clinical studies, we're talking about tens of people today, are in effect cured. Their hemoglobin levels have returned to normal and they're as healthy as everybody around this table in terms of their ability to walk, to move around. In another area, Intellia Therapeutics is working on a disease called transthyretin amyloidosis, where they're looking at deleting a gene in the liver to cure that disease. The first phase 1 clinical studies are in progress for that project. They're able to deliver CRISPR to the liver, and this picks up on the point that Sharon said earlier on. If you want to correct a gene in the body, it's very easy to deliver a gene to the liver. At least it's technically, we know how to do that. Those first patients on that study, in essence, they show a 90% reduction in the level of expression of that mutant gene. And it's still very early days, but hopefully that will manifest itself as a decrease in the symptoms of that disease. So we're really excited about applying that, well, the field is very excited about applying that technology across a range of different diseases. And the early diseases that will be treated using CRISPR, they're diseases in the liver, they're hematopoietic stem cell diseases, blood diseases, hematologies, they're diseases in the eye. And as Sharon says, whilst we work at creating the technology that allows us to deliver CRISPR to other tissues, such as the brain or the kidney or the lung. Maybe I'll stop there.
No, that's fantastic. I could listen to you all day, but I'm really interested in this stuff. But there's also another bit of jargon, which would be great for you to go into, Steve, on a similar vein, which is AAV medicines. What's that all about, and how does that differ from other approaches?
So AAV stands for adeno-associated virus. It's a virus that the scientists are using to deliver genes to cells in the body. And again, AAV, if you deliver AAV to a patient, to a human being, it will go to certain tissues. And again, one of the primary tissues is the liver, maybe some to the heart, maybe some to the brain. And what we're able to do using AAV is if a patient has a genetic defect, so a mutant gene, we can use AAV to replace that gene. So the difference between the two is what CRISPR does is it allows you to correct a gene in the DNA of the body such that you repair the gene. And you, in essence, take a mutant gene and repair it to normal. What you do with AAV is you add an additional copy of the gene to a cell such that you can begin to express a functional protein. And again, AAV therapies are in the clinic. A medicine launched this year by Roche called Zolgensma, which treats spinal muscular atrophy. Second medicine called Luxturna, which treats an inherited retinal disease by, again, adding a new copy of the gene to cells in the patient. So again, very early days, but through a combination of the two technologies, we can add additional genes to cells in cases where we're replacing a gene that is no longer working, or using CRISPR, we can repair a gene, or indeed, for that matter, delete a gene to lead to efficacy in a disease.
Wonderful. Thanks so much, Steve. Now, it's very apparent that AstraZeneca has made a very big, bold move into the rare disease landscape with its collaboration, its marriage with Alexion. Alexion has been there already. So it's clear that this is important to both companies, clearly. But I was just wondering, what would you like to see over and above this? What else could happen other than two of the most innovative large pharmaceutical companies in the world, focusing on this area? What else could expedite the delivery of medicines to sufferers? Sharon, is that something you're okay with taking?
Sure. So Steve and I both sit in the research space because that's the part that we love. But we can both tell you that drug discovery and development is part of a whole ecosystem. And bringing together AstraZeneca and Alexion really helps us improve that ecosystem. So it starts at the beginning with, what I call a good idea, that new target identification, and then how we're going to go about addressing that. So the first step is that we no longer have limitations on how we're going to address that. Whether we want a small molecule or a large molecule or an enzyme replacement therapy or genomic medicine, all of those things are options. So we can fit the right therapy to the disease. We can build a therapy that the patients most need. And I think that's enormously valuable. But then we have to get that into clinical trials. And running rare disease trials is a pretty unique setup as compared to some other parts of drug development. Alexion has really built our expertise in that space, and we get to bring that to AstraZeneca. But once we move beyond those early stage clinical trials and we're trying to reach a global population, AstraZeneca has a larger reach than Alexion has. And so there is that wonderful opportunity to be able to make our medicines accessible to more patients worldwide. So I think if we start from the beginning, we have every technological opportunity in front of us. We don't see limitations. And I think that's incredibly exciting as a scientist. But the second part that makes, I think, our job valuable is that we're not just engaging in science, but we're creating therapies for patients. And what matters to us scientists is knowing that those therapies will get to the clinic and get to the patients. And this acquisition of Alexion by AstraZeneca expands that reach enormously. And it increases our confidence in our ability to get those new ideas all the way through to a therapy for patients in a time frame that's meaningful for them.
Great. Sharon, thank you so much for that explanation. We're now going to have a bit of Q&A, the last minutes at the end. We've actually had one in the group chat, which is quite timely given what Sharon was just talking about in terms of the new capabilities and broadening that out to different parts of the body and different tools to address different diseases like we haven't had before. So I'm just going to read this out. It's you mentioned that delivering therapies to the brain is not possible today, but will be in the future. Can you estimate a time frame that such therapies may be readily available? Sharon, I think that's probably yours.
So first, I wouldn't say it's not possible. I would just say that we haven't nailed it yet. The brain is a complex system, and you have to be able to deliver your drug, not just to the brain, but to the right part of the brain. And so I think that there are lots of endeavors in play right now. I just don't think we've perfected the system. How long will it take? That's a tough question to answer. Given that there is work underway already and that we're striving to understand the best delivery techniques, both in terms of the construction of the molecule and in terms of the delivery of it, I would say within a decade feels like a safe bet to me. What would you say, Steve?
I think that's a pretty safe bet. There are many, many organizations, including us, attempting to solve this problem. And we do have one project running internally, which would involve delivery to the brain. So I think, yeah, comfortably within a decade.
Great, thank you. That's that's wonderful news certainly from where we are just a few years ago. Indeed, if I've got my dates right, we had the first gene therapy approved a couple of years ago in America, and there was a lot of excitement then. It's why we're largely why we're here today and talking about it. But more recently, the news flow on the drug approval side of things hasn't been as good. And there might be people out there who, who concerned that whilst the scientists are doing their bit, maybe the regulators aren't doing theirs, or have got a different agenda. I was just wondering, Steve, maybe could you take this one? What's your view on that? What's going on, and what's the outlook?
Well, I think the outlook, I think, is very positive. But then again, I'm an optimistic person, and I believe that within what we do in early research, we can solve big problems. That's why we're here. Much of the issues with these medicines are the potential safety concerns around these medicines. And for both CRISPR and AAV gene therapy, there are two primary safety issues. The first is an immune reaction when the medicine is given, but that by and large can be managed. The second might be, what do these medicines do to other parts of the genome, other genes in the body that you don't necessarily want to target? So there's a risk with CRISPR that it will delete the gene you want, and it might delete things elsewhere. There's a risk with AAV, different risk, that AAV will manipulate other parts of the genome. And those are the reasons why many of these studies have been stopped, because of concerns around, ultimately, it would be concerns that these medicines affect the genome and you generate a cancer. Those are the concerns. A huge amount of work is ongoing to minimize the risk of that happening and understand why it happens. But also, I think something like with any medicine, we need to consider the risk versus the benefit. We've all been obviously experienced the pandemic over the last 18 months. There are risks with the there's risks with all the vaccines. The ones that most of us know about are the risks with the AstraZeneca vaccine. Those risks are 1 in a million, 1 in 10 million. So we need to understand the safety risks associated with medicines. We need to weigh them up against the benefits, and then we may need to make an educated decision as to whether the potential benefit of taking the medicine outweighs the risk. But that is no different with the gene therapy to any other medicine, including the likes of aspirin and all of these things that we take over the counter today. So I think once we understand those risks on a disease by disease basis, we will be able to proceed with taking these medicines into the clinic for the treatment of many of these diseases. And the types of things that will stop us, there's a question here about the ALMS1 gene, Alström syndrome. I don't know the details of that disease, but just to put some perspective on the question here is, we've been told that this gene is too large for gene therapy or repair. Now, the specific issue there, and I don't know how big that gene is, but using AAV that I was talking about, you can only put about 5,000 bases of genetic material into an AAV. If you try to put more than that into an AAV, it's too big and it doesn't work. So I'm assuming that's the problem with that gene. In 5, 10, 15 years' time, there will be other approaches that will be developed that may well solve that problem. I don't know what they are today, but as I say, CRISPR didn't exist 9 years ago, and now it's in the clinic treating disease. I think we can be optimistic that these problems will be addressed as time goes by.
Great, thanks, Steve. Sorry, Sharon, go for it.
I was just going to jump in about the panelist question about Alström syndrome. I think that really beautifully illustrates the complexity of rare disease. So can we make a genomic medicine for ALMS1 mutations? It's complicated because there are hundreds of mutations that are associated with disease, and we can't fix them one at a time. And so it sort of depends on where they're localized in the gene and whether or not it's possible to address a group of them. That said, as I understand it, Alström syndrome is a ciliopathy, and there are other academic groups that are understanding whether or not there's a small molecule that might help address ciliopathies. So as we think about rare diseases, we think about all the aspects of the disease and the best way to impact that disease and what kind of therapy we need to build. So today we're talking about genomic medicines, but it's not the only answer for patients with rare disease.
Thank you. We've got loads of questions and we're coming up against time. I'm going to try and just sneak in one and hopefully there can be some follow-up. We'll all be at the networking site for the conference afterwards, so there can be follow-up questions then. But one that I think is interesting is moving a little bit away from the CRISPR side of things in terms of specific genetic deletions. And I'll just read it straight from the chat, which is, what is the case in conditions with chromosomal deletions, duplications, etc., where there are much bigger issues and at play? So what's going on there?
So from a scientific perspective, this is orders of magnitude more challenging. several orders of magnitude more challenging. And I think it's fair to say that today the science is not there to address diseases that are caused by these larger chromosomal deletions. Maybe that will come with time. Sharon was talking about maybe 10 years for brain delivery earlier on. Unfortunately, I suspect this will be longer.
Yeah, it's unfortunately a very scientifically complicated problem. Sometimes, in the case of a translocation, you get the break and the interruption of a specific gene, and then maybe in certain cases that could be addressable. But for the deletion of an entire chromosome or the duplication of an entire chromosome, we don't have the technology to address that today. I wish we did.
Well, firstly, I would like to apologize to those online who've written some fantastic questions. We're going to have to end the session now. There's plenty to talk about. It's a really interesting subject, and you can hear the enthusiasm from Steve and Sharon in terms of them working together and what AstraZeneca and Alexion are hoping to do over the next few years. From my side of things, thank you all for joining in. For those of you who do want to continue the conversation, we can go to the other venue in the conference. I'll be happy to talk to you there. And thank you again, Steve and Sharon, for your time. And yeah, waiting excitedly in anticipation for what you guys are going to be coming out with.
Thanks for the invitation. It's been my great pleasure. Thank you.
Likewise, really enjoyed the conversation. Thank you.